Hello, today I will be presenting the design and validation of mammalian expression vector for CAP2A1. CAP2A1 is a liver enzyme involved in the metabolism of alcohol, drugs, and environmental toxins. Its expression induced during xenobiotic exposure and elevated CAP2A1 levels have been associated with liver cell damage. In this project, CAP2A1 is overexpressed in liver-derived mammalian cells to investigate the cellular effects of increased CAP2A1 expression. As shown on the slide, I found CAP2A1 protein sequence in UniProt KB, then I reverse translate the protein sequence to cDNA sequence, which can be used to design a synthetic gene for cloning into mammalian expression vector. In the expression plasmid, I choose the pcDNA tree expression plasmid because it's designed for high-level gene expression in mammalian cells. This vector contains CMV promoter, which enables strong expression of the inserted gene in human cell lines such as HepG2. In addition, pcDNA includes a bacterial origin of replication and the antibiotic resistance gene, allowing the plasmid to propagate and amplified in the E. coli before transfection into the mammalian cells. This makes pcDNA suitable for cloning and subsequent expression of the human CAP2A1 gene. Before sending the gene of interest to synthesis, several modifications are required. Restriction enzyme sites are added to both 5' and 3' ends of the CAP2A1 cDNA selected based on the multiple cloning site of the plasmid. Using two different restriction enzymes ensure directional cloning and correct orientation of the gene within the vector. At the 5' end, Kozak sequencing added to enable efficient translation into mammalian cells. At the 3' end, his tag and stop codon are added to purification and proper termination of translation. After these modifications, the synthetic gene is ligated into the vector. The plasmid is first propagated in E. coli and then transfected in the human liver cell derived cell line to express CAP2A1 protein. All of these summarized into the diagram. The first figure, you can see the full CAP2A1 gene designed for synthesis. I selected ACC65I for the 5' end, XY for the 3' end. The restriction sites are not present within the CAP2A1 gene, which allows clean insertion without cutting the gene of interest. Using two different restriction enzymes ensure directional cloning, so gene is inserted into the MSC in correct orientation. Additional elements such as Kozak sequences and his tag and stop codon were included. After synthetic gene is produced by a company, both insert and the vector are digested using the same restriction enzyme we added to form the sticky ends. At this stage, plasmid is dephosphorylated to prevent ligation. The correct DNA fragments are then selected by agarose gel electrophoresis, purified, and finally generate recombinant DNA construct. As shown in this figure below, the strategy ensures correct insertion and orientation of the gene within the vector. After ligation, the recombinant plasmid is introduced into the component E. coli cells by transformation. The transformed bacteria are plated on agar-containing ampicillin, so only cells that have taken up plasmids can grow. Because not all plasmids necessarily contain gene of interest, bacterial colonies are screened to identify correct clones. Individual colonies are picked into the mini cultures and plasmid DNA is isolated during using a mini-prep procedure. The extracted plasmids are digested within the selected restriction enzymes and analyzed by agarose gel electrophoresis. Plasmid containing gene of interest show two bands on the gel, the vector and the gene of interest, and where the empty vector show only single band. Further confirm the correct construct, plasmid DNA from positive clones is analyzed by Sanger sequencing. The sequencing is performed using a primer that binds within the CMV promoter. In the final step, verified recombinant plasmid is transfected into the HepG2 cells for protein expression. CAP2A1 expression is confirmed by Western blot using anti-CAP2A1 or anti-His antibody, and enzymatic activity assays can be used to assess functional expression. The His tags enables affinity purification of the recombinant protein. Together, these steps confirm successful expression and purification of the CAP2A1 in a human liver cell lines. Thank you for listening to me.